Welcome to this explainer. Today, we're diving into one of the most wild and universal things happening in biology right now, how our cells read their genetic blueprints to build the proteins that keep us alive. It's a highly conserved mechanism across all life forms. Yet, the strategies used to pull it off can be radically different depending on the type of cell. I am so excited to break this down for you, so let's just jump right in.
So here's the big question. If literally all life on Earth, from a tiny microbe to you and me, relies on the exact same fundamental genetic code, why do our cells process that information so differently compared to, say, a bacteria? It's kind of a huge mystery of cellular evolution. Today we're going to explore this by comparing the fast-paced, scrappy bacterial startup approach to the highly regulated, compartmentalized eukaryotic corporation.
Here's a quick roadmap for this explainer. We'll start with the universal protein blueprint, then look at the speedy bacterial startup versus the secure eukaryotic factory. We'll weigh the specific evolutionary advantages of both strategies and wrap it all up with a summary. Let's get to it.
All right, Section 1, the universal protein blueprint.
Before we get into the differences, let's just ground ourselves in what every living thing has in common. It comes down to two highly conserved steps. First up, transcription, copying that DNA master blueprint into a working mRNA document. Then translation, where ribosomes decode that RNA to build the actual protein product. Bacteria, humans, plants, we all rely on these exact same two steps to express our genes.
But this is exactly where the paths completely diverge. The absolute biggest game changer when comparing a prokaryotic bacterial cell to a eukaryotic cell is the nucleus. Our eukaryotic cells have a distinct membrane-bound nucleus locking up that DNA. Prokaryotes don't. Their DNA just kind of hangs out right there in the cytoplasm. And as you'll see, that one single physical barrier, or lack of it, basically changes everything.
Moving on to Section 2, Bacteria, the speedy startup.
Think of bacteria as these incredibly agile, fast-moving startups where everything happens in one giant open-plan office. Because there's no nuclear membrane separating the DNA from the rest of the cell, there's zero physical barrier between copying the blueprint and building the product. This creates something amazing called coupled transcription and translation. Literally, the second an RNA polymerase starts transcribing a piece of mRNA, ribosomes just instantly latch onto that emerging RNA strand and start building the protein. It's happening simultaneously, right there in the same cellular compartment.
To make things even more efficient, bacteria use polycistronic mRNA. They link genes for a specific job together in what's called an operon, like the TRP operon for synthesizing tryptophan. Instead of printing out separate instruction manuals for every single tiny part, the bacteria just transcribe one massive, super efficient master manual. Ribosomes then translate that into multiple related proteins all at once. No wasted time. Now, let's contrast that in Section 3, Eukaryotes, the secure factory.
If bacteria are that scrappy, single-room startup, eukaryotic cells, like the ones inside us, are massive, highly regulated corporations. The executive office is strictly separated from the factory floor by the nuclear envelope. So transcription and translation, they absolutely cannot be coupled. First, DNA gets transcribed safely inside the nucleus. Second, that raw RNA gets heavily processed. Third, it has to be physically shipped out into the cytoplasm, and only then, in step 4, can ribosomes finally get their hands on it to start translating. What's really fascinating here is the intense level of security and quality control that happens before that RNA is ever allowed to leave the nucleus. We're talking serious processing. Introns, the non-coding regions, are spliced out. And notice those exon junction complexes added to the splice sites? That's a crucial checkpoint. It literally tells the cell, hey, this transcript is fully processed, so half-baked RNA doesn't accidentally leak out. Toss on a 5' cap and a poly A tail for stability, and every single step is rigorously checked off before shipping.
Which brings us to Section 4, the bacterial advantage.
Now, you might be wondering, if the eukaryotic system is so incredibly secure, why would any cell stick with the bacterial method? Well, it all boils down to raw energy and efficiency. Bacteria have tiny genomes, maybe 500 to 4,000 genes, and unlike us, they don't have genomes clogged up with repetitive sequences. Their DNA mostly codes straight for proteins. By keeping the genome small, running everything in one compartment, and totally skipping the whole RNA transport nightmare, bacteria waste absolutely zero energy.
So this coupled system is just built for pure unparalleled speed.
Because they skip processing and transport, multiple ribosomes can pile onto the mRNA while it's literally still being transcribed. Plus, this coupling gives them wild rapid response tricks, like attenuation. Because translation is happening right on the tail of transcription, if a bacteria suddenly senses it doesn't need a specific protein anymore, it can halt transcription mid-process. Boom, instantly stopped. It's real-time adaptation at its finest.
Moving right along to Section 5, the eukaryotic advantage. If the bacterial way is so insanely fast and efficient, why on earth did eukaryotic cells spend so much evolutionary energy building a complex nucleus? Well, what we sacrifice in startup speed, we make up for it with advanced cellular control. Segmented processing, keeping transcriptions safely locked away while translation happens outside, lets our cells tune gene expression in ways bacteria can only dream of. We can pack our DNA tightly into chromatin using histones to restrict access, and we meticulously control exactly when and where the mRNA goes. This separation is basically the secret sauce for multicellular life. Because of alternative splicing, a single eukaryotic gene can actually produce a bunch of different, finely tuned proteins just depending on how the cell decides to process the mRNA. It lets us react to highly complex physiological and developmental cues. And once translation is finally done, we have these advanced trafficking systems to ship specific proteins to exact locations within the cell. It's the ultimate power of control.
All right, wrapping things up with Section 6, Summary, 2 winning strategies.
Let's bring this all together. When we look at location, bacteria do everything right there in the same compartment, the cytosol. Meanwhile, eukaryotes split the work between the nucleus and the cytoplasm. That location difference fundamentally alters the timing. Bacterial processes are completely simultaneous and coupled, while eukaryotic processes are strictly separated in time and space. So the ultimate core advantage? For bacteria, it's blistering speed and metabolic efficiency. For eukaryotics, it's the capacity for complex regulation and rigorous quality control. Both are absolute winners in the evolutionary game.
I want to leave you with a thought to chew on. We've seen how just one structural change, adding a nuclear envelope, completely revolutionized how life reads its genetic code. So, as these highly conserved molecular machines keep adapting over the next few million years, what entirely new evolutionary strategies might life invent next? It is definitely something wild to ponder. Thanks so much for hanging out with me for this explainer. Keep learning, and I'll see you next time.